Sir, good morning, sir.
Good morning. How are you?
I'm fine. Thank you, sir.
Um,
The- If any problem arises, we share, sir. Kindly share your PPT to my WhatsApp.
yeah, I'll.
If any problem arises, from here we will share.
Um,
Yeah, visible and audible.
oh, oh, Typically, I go to the office. I guess
Okay.
it's a conference room,
Yes, sir.
but it's mark almost 11, 12 आता ఉంది.
Okay, okay.
Mm. I'm doing it from my home.
Okay. Please unshare, sir. Whenever I tell you, just share it.
I did share that file.
Hmm.
You want me to send an email or
Just the WhatsApp, you send through my WhatsApp or mail.
WhatsApp? I don't have that one.
Okay.
It's a picture shot kind of thing. It may be a little bit difficult to send it to
Mail,
that
mail,
one.
or you mail also you can share.
If this is okay from my side, it's no problem. We have faster internet.
Okay.
Can you see this one?
Yeah, yeah, visible.
Okay, then there won't be any problem.
Okay,
From my side, there won't be any problem.
okay,
Otherwise, I can't even send this one through email because it has a larger file. Maybe I have to compress and do it.
okay,
It has some pictures and kind of things, so that's the reason it is having a bigger size.
okay, sir, no problem. Okay, sir, you unshare it. Whenever I tell you, you share it.
Okay. Mm, uh, I typically don't use the Zoom that much. We use WhatsApp,
Meet, sir, Google Meet.
Google Stream. But our university has WebEx has a license for it, so we use that one
Okay,
for classroom instruction. I do teach one course. Um, that even distant students are there from all over the country. They join. We have an online master's and PhD program.
sir.
Our university started a couple of years back online master's and PhD program. If somebody is working, they can. Only thing is they have to pay the tuition fees, but they can work and the respective universities,
Yes, sir.
and then get a degree from here.
Okay.
So for that I use for Zoom instead of Zoom. Zoom we don't have. Some people have it, but I don't have the license for Zoom.
Okay, okay.
Yeah, so log into your account. It's not a problem as long as otherwise it has only 40 minutes or something.
Okay.
So I'm also planning to uh, planning to retire this next month.
Yeah, I'm going to retire, sir.
Um, Next month, 30th. So I've been here at Mississippi State for 37 years.
37 years.
Yeah, I'm getting by June, July, I will be 70, 72 also.
Sir. Their retirement age is 72, sir.
No, no, there is no retirement age. You can work as long as you wish.
Okay.
Um, but people typically retire after 65.
Okay.
When they reach 65, they typically retire because they want to do other things,
Okay.
just not working. Our department has horticulture, agronomy, crop science, soils, um, weed science together.
Okay.
So all the branches are together in my department.
Yes, sir.
It's one of the largest one with, I think it's a 38 faculty on campus.
Okay.
I think we did, I probably, I heard it from one of my former postdoc. His name is, he's from, I guess, former AP Agriculture University student, Bapatla. He's from Bapatla. His name is Gopal Khakani. I don't know whether he met you. He might have been to Bapatla College, but I'm not sure.
Name, sir. Name.
Gopal Khakani is a professor at University of Florida currently.
Uh,
Um, um, he worked in my lab for about 5, 6 years as a postdoc.
Kakani Ramesh, sir.
Hmm?
Kakani Ramesh,
Uh, no, Ramesh is
um,
he's at another university. He's our student. Yeah, PhD from here. So how long have you been here?
Myself, sir,
How long have you been working?
that 27 years, sir.
27 years. Oh, okay.
Before that, Injiranga, Agriculture University. After
Okay.
state bifurcation, this and 2007-8, this Horticulture University was established. And separate
2007.
Agriculture University and a Veterinary University. So three universities came into
Yeah.
Mm.
I have been to veterinary university one time. Our administrators came. It's been a while, but
Tirupati, sir.
mm, we did not come to any horticulture universities. But AP
So,
Agriculture University, we went there
this
at
is
that time.
near Tadepalligudem. That is Vijayawada, Yelur, Tadepalligudem.
Yeah,
So 100 kilometers from Vijayawada.
100. Okay, not
Rajahmundry.
too. So, at your location, what are the major horticulture crops that you focus on?
Here coconut, banana, some vegetables
Okay,
and cocoa.
vegetable crops. Okay. Here we have ornamental horticulture crops. Poinsettia, has some kind of things, and the main horticulture crop that our people work is on both tomatoes, indoor and not outdoor, but indoor
Okay,
mm, greenhouse, hoop houses kind of thing. And then one of the major crop is sweet potatoes.
okay.
And then they do have like cowpeas and other things. So cowpeas are also main crop. Means not major crop, but it's one of the crop they produce for local markets.
All crops will be there here, but major dominating coconut, banana, oil palm, vegetable.
Okay.
Some
So here, because of the climate,
so,
we cannot produce any bananas. So they won't flower. Or
it has a tropical fruit, na banana.
yeah, so no bananas, no. I guess coconuts. Only had to go to the coastline, Florida or somewhere. So do you have both PhD and master's programs?
Yeah, yes, sir. MSc
Okay.
and PhD. MSc and post graduation at two places. So here at Viyargudem, that is university headquartered. Another is Anantarajpeta, Kadapa district.
Okay.
College
Yeah,
is
I've
so
been there. While I was working at SV University, we visited that one couple of times.
so
Uh,
in Anantarajpeta, sir.
yeah,
Anantarajpeta, railway corridor.
yeah, railway corridor. Yeah, it's close to Tirupati. Then
Yeah.
not too far away, but uh, at that time we were taking MS students on a botanical tour.
Okay.
So for that reason, one time we went there.
Okay. Okay.
Um,
Visit our
attending.
university, sir. Once,
Hmm.
whenever you come to Andhra, please visit
Yeah,
our university also.
yeah, I have several. One of my former postdoc, he is at Orissa State University, Bhubaneswar. He is there. He is from Andhra, but he is posted the state one of the research programs. And then another visiting scientist came from Bhubaneswar. Um, he is also there, but I don't think I have known that many from Sarkar areas. Very few of them. I came from non-agriculture university, so that's one. I
In
visited them,
Tirupati,
but in Tirupati and Hyderabad. But
that our native place is Pileru, that village, what is the name of village?
it's called Marella, Marella.
Moral,
Uh, Marella. By
Moral.
surprisingly, there is an Italian company that started by name called Marella. There is one village in, I think it's in Ongole also, it's the same name.
Okay.
It's a small village, about 30, 45, 50 miles from Tirupati. It's on Tirupati Kadapa road.
Yeah.
It's a very remote village. It's on the backside of Tirupati Hills.
Mm.
So there is no connection. It's cut off from last
So,
village.
37 years back, you went to America. It is a very great, sir. And you have excellent career record, so many awards. Really, we are very happy to be here as a guest speaker today and give lecture, guest lecture to our students, teachers, scientists, all 44 institutes, the people will participate today.
Yeah, I had pretty successful program over the years at Mississippi State. I worked with really talented people. Means my program has a lot of people that came and contributed. It's not by myself, but there are quite a number of people came.
Recently, MS Reddy garu visited, sir, our university, MS Reddy, Shankar Reddy garu, PGPR.
Oh, MS Reddy, yeah, I know. He's in Auburn. Next to it's about 3 hours drive from me.
Okay.
Yeah, he visited recently, right? Last a couple of months back, maybe.
We have signed MOU also with PGPR,
Yeah.
and he participated in international workshop, international seminar.
Okay. Yeah, he's active, still active. He's retired, but yeah, still active.
Yes,
He
sir.
has
Yes.
some business and other. That is a chair. Found. I guess is a founder of that society.
Yeah.
Yeah, as you all know, that has a WhatsApp group that has a lots of messages comes back and forth.
K. Ramesh,
uh, MS Reddy, yeah.
K. Ramesh Reddy. That,
Ramesh,
um, sir.
Ramesh Reddy.
Yes, sir. Umesh
He's
Reddy.
in Florida.
Umesh Reddy.
Umesh Reddy, we know. Yeah, yeah. He came to my home also. He worked for some time in Mississippi for a couple of years.
Yeah.
From here, he went to West Virginia.
West Virginia State University is working presently.
Yeah, yeah. So we worked together on certain projects. He did some molecular work on some of the material that we worked, sweet potatoes. Um, Non-published, one or two papers we
Yes,
had
sir.
common papers. I know him, he and his wife, he came to our house, visited our place. He used to work about 145 miles to my place. It's another small university at that time. He was a faculty there. I think he suggested, I think he might have suggested you guys about my,
Yes, sir. Yes, sir. He
and
was the speaker for this Global Connect program.
yeah,
With him, we start.
there is another postdoc. Um, he he's the one who I think there are a couple of contacts there. Amarnath Reddy, Venupus,
Yes, yes, sir. Amarnath Reddy also.
yeah. So he's the one. I just talked with him. He's on the East Coast. He's 12:30 now for him. So how many students are there in the whole campus?
Campus, I think in Yergudem campus, 1,800 like that will be there. So
Hmm.
totally five colleges are there, sir. Yergudem, Anantarajpeta, and
Okay.
Parvatipuram, Pulivendula. So in affiliated college also we have.
So good morning, sir.
Oh,
Good morning, good morning.
This is Dr. Nagaraj, sir, from College of Horticulture, Pulivendula. Associate.
Dean College of Horticulture, Pulivendula, sir.
oh, okay.
Dr. Nagaraj, Associate Dean.
Sir, this is new college, sir. Pulivendula is new college. Here we have two batches only, first year and second year. In first year only 47 students, and second year, first year 60 students, sir, and second year 47 students.
Oh, okay. It's a
Not students.
totally new one.
Total students now it's for them summer vacation, and they are joining through online, sir.
Okay, okay. Our campus is not really big. We have 24,000 students on
54,000
campus. 24,000 students. And then the city population is only 28,000. So altogether it's about 50,000, small town.
So most of the students, sir.
Beautiful campus, very spread out and very nicely maintained campus. And then,
Radha, sir.
oh, good morning. How are you? I thought you were Dr. Shastri. He came as a visiting scholar from India to our lab. We spent about a year almost. He's at Warangal. I sent him the link, so he joined there from there.
Okay.
So very good morning, Professor.
Good morning, sir.
Yeah, nice to see you, sir, again. Yeah, Professor Reddy is my mentor and my well-wisher,
Okay.
and I'm totally changed based on the Reddy motivations and support. I have never forget the Reddy support.
Excellent, sir.
Yeah,
Now,
at that time we had
yeah,
four, five came from India at that time.
yeah,
Welcome to ISR Horticulture University, sir.
yeah, very nice, sir.
My Vice Chancellor, Dr. K. Gopal, sir.
Yeah. Is he here or he said he's in Delhi or
Delhi,
something?
sir. Sir is in Delhi, and Register Dr. B. Srinivasan and our Vice Chancellor sir both went to Delhi 2 days back.
Okay.
If time permits, they will join also. So people are joining, sir. We will start within 5 minutes. Sir.
Okay. So everything is online or you have some at your place?
Different online, sir, throughout the state.
Okay.
This all 44 institutions located in different places. Rayalaseema and coastal Andhra, central coastal.
Okay. Okay.
Signals okay, sir. Here rainy.
Oh,
No problem, no signals.
from my side, there is no problem. We have pretty good internet connection at home. It's almost better than at office.
So just I'm suffering some cold and fever. My voice may not be that much clear. Please excuse me.
That's fine.
Good evening, sir. I'm from Amar from Delaware State University.
Good morning, sir. Here, thank you. Welcome to Dr. YSR Horticulture University. Thank you for
Thank you.
participating.
Thank you. Good to see both of you, Dr. Reddy and you, sir. And I'm happy to join this meeting again.
Thank you. Sir Raja Reddy garu, shall we start?
Yeah, sure.
Good morning. Welcome to Dr. YSR Horticulture University, Venkataramanagudem, West Godavari District, Andhra Pradesh, for the 24th Global Connect series in collaboration with Mississippi State University, USA, on climate change and crop productivity, facilities, tools, challenges, and opportunities. And today, the program chairman, our Honorable Vice Chancellor, Dr. K. Gopal Garu, and with his visionary leadership and dynamic leadership, we are conducting 24th Global Connect series. Thank you, sir. So, sir is in busy. Due to that, he may not join. And with his leadership, we are conducting 24th Global Connect series, almost 2 years over. And this is to update the knowledge of different topics, different areas in horticulture by the eminent scientists throughout the world. And this is the 24th Global Connect series. And especially to update the knowledge to the students, teachers, scientists, and faculty from the different horticulture research stations, colleges, polytechnic colleges, and KVKs, and all 44 institutes are working in Dr. YSR Horticulture University. Thank you, sir. Honorable Vice Chancellor, Dr. K. Gopal Garu, for giving the permission to conduct this 24th Global Connect series. And today, guest speaker, Dr. K. Raja Reddy Garu, William Gills, Distinguished Professor, Mississippi State University, Department of Plant and Soil Sciences, Mississippi State, USA. Welcome, sir. Welcome to this Global Connect series. Thank you for accepting as a guest speaker today. And our Dean of Horticulture, Dr. Norman Naidu Garu, Director of Research in Charge and PG Dean, Dr. K.T. Venkataraman Garu, Director of Extension, Dr. Govindarajan Garu, Dean of Student Affairs, Dr. Suri Kumar Garu, Registrar, Dr. B. Srinivasan Garu, Controller of Exams, Dr. Prasanna Kumar Garu, University Librarian, Dr. Ramanandan Garu, Controller, Dr. Srinivasan Garu, Estate Officer, Ramana Garu, all Associate Deans of Colleges of Horticulture, all Associate Directors of Research, Coastal Zone 1, 2, 3, and all principals of polytechnic colleges, all heads of research stations and KVKs, and all students, scientists, teachers of Dr. YSR Horticulture University. Welcome to this Global Connect series. And today, Dr. C.H. Nalini, Assistant Professor, Entomology, College of Horticulture, VR Gudem, and Dr. S.B. Ramya Lakshmi, Assistant Professor, Agriculture Economics, College of Horticulture, Anantarajpeta, will act as a rapporteurs today. And before going to lecture by our respected guest speaker, just a brief introduction about our guest speaker. Dr. K. Raja Reddy Garu earned his BSc and MSc and PhD degrees from Sri Venkateswara University, Tirupati, India in 1975, 77 and 84. Sir is native of Morella, near Pileru, nearby Tirupati. Sir is 37 years back. He went to USA and presently working as joined as a plant and soil science department at Mississippi State in 1988 and became a research professor in 1992. He was named as William Gills Distinguished Professor in 2021. Dr. Reddy's research focus on the effect of anthropogenic climate change, remote sensing, and crop modeling applications on agriculture resource management through the lens of environmental plant physiology. He has over 37 years of research experience in Mississippi State and manages the state-of-the-art sunlit plant growth chambers known as the soil plant atmosphere research. He is credited with numerous significant discoveries in various areas of agriculture. His research topics include the impact of climate change on crop physiology and the growth, and the development of several essential crops such as cotton, soybean, rice, corn, sorghum, sweet potato, and switchgrass, as well as the native grasslands and forage crops of global importance. Additionally, he specialized in remote sensing, stress physiology, and crop model applications. Dr. Reddy has authored over 350 publications, including 2 books, 4 edited volumes, and more than 32 book chapters. In addition to his research responsibilities, he created a capstone graduate level course in environmental plant physiology that integrates research, teaching, and learning based on his work using the advanced SPARE facilities at Mississippi State. He has secured external funding from federal, state, and industry sources to support his research and training programs. And Dr. Reddy has received numerous recognitions and awards, including L.R. Ahuja Agriculture Systems Modeling Award 2023 from Soil Science Society of America and Faculty Achievement Award 2019, Raphael E. Power Award at Mississippi State 2012, the ICAC Cotton Research of the Year Award 2020, and the Outstanding Research Award in Cotton Physiology 2010 from National Cotton Council of America. He also received the Outstanding Contributions to Science Award from the Mississippi Academy of Sciences 2020, the Superior Faculty Research Award 2018, and the International Service Award 2016 from DAFE, as well as the Excellence in Research Award from MAFES 2006-2018 and 6 MAFES Outstanding Publication Awards 2002, 2006, 2012, 14, and 21. In recognition of his contributions, he was elected as Fellow of the American Society of Agronomy 2005, the Crop Science Society of America 2006, the American Association for the Advancement of Science 2020, and the Mississippi Academy of Science 2021. He was also recognized in 2020 as one of the world's top 2% scientists in biology by Stanford University. Dr. Reddy has trained over 40 visiting scientists and 16 postdoctoral research, along with 26 graduates, 16 PhD, and 14 MS, and 11 undergraduate research scholars. His trainees come from diverse background and focus on areas of such as stress, physiology, climate change, crop modeling, remote sensing, and global security. So it takes a lot of time. Sir is a very eminent, excellent scientist in the world. So congratulations, sir. Congratulations, and thank you very much for accepting as a guest speaker today. And this is just a brief biodata. To read, it takes a lot of time. And thank you very much, sir, once again. Please proceed.
First of all, thank you very much for the invitation. It came from multiple people suggested to provide that lecture series. So finally it happened. I personally thank you for making all the arrangements, and all your administrators for making this one happen. And also it gives an immense pleasure to talk with our own people coming from back home, from the state of Andhra Pradesh. So I'm the native son of the state. So today I wanted to share some of the work that I've been doing. It may be not totally horticulture, but it's kind of, I work across many species that include horticulture crops. The focus will be on fruit production. That's kind of its challenges for lots of things, but its focus should be on that crops and food and people kind of areas. Before I go to the presentation, Mississippi State University is, we have about 24,000 students on campus. And then we have about 1,400 faculty. 1,400 faculty. The resident population is about 28,000. So it's not like a big town, it's a college town. So that's where I came from, a very small village. So it's nicely fits into my thinking and also my kind of upbringing as well. So let me share my talk and then we go from there.
Yes, sir. Please, sir. Put it in slide mode, sir.
So you can
Yeah.
see that.
Yes, sir. Yes, sir. Okay. Proceed.
Okay. First of all, again, thank you for the opportunity to talk with you, all the faculty, students, and administrators at the Dr. YSR Horticulture University. Our department has all these like weed science, crop science, soils, and that also includes horticulture crops, horticulture discipline also. So it's an integrated department. We have about 38 faculty on campus and another 20 faculty off campus working at different experimental stations. So to start with, I'm going to be talking about climate change and crop productivity, facilities, tools, and challenges as challenges and opportunities. So to give a little bit of background, I'm from Marala, which is a very small village, about 50 miles 50 miles from Tirupati. It's a very small village. It's kind of almost like a end of the town at the backdrop of the Tirupati hills. So there's no connection beyond that side. So even now they don't have a connection going that one. We have to go to either Kadapa or come to Tirupati or Chittoor, which is a district headquarters. So I'm from here. My family, all of them. I have 4 brothers, 2 sisters. Everybody is back home. They do mostly. We worked on agriculture crops. So my family is from agriculture family. So we work on multiple crops, including horticulture crops. That includes, I guess, particularly we are looking. Vegetable crops, but we also look at mangoes also. And this is where my 1 to 5 grade started. There was no building, but that's what that my 1 to 5 grades, that's where my early education started under this tree. Then we moved to some kind of away from 6th grade to 10th grade, some JP high school, a little bit away from that one. This is where I came from. So I got the schooling at one of the pretty good school at that time. That's where I learned English alphabets in 6th grade. And then I went to, after the school, I went to, we are the first batch of SSC, first batch of SSC. Then I went to intermediate, which is very close to your place, maybe Gudur, which is not too far away, but it's Nellore district current at that time. And then 1975, I got botany, zoology, and chemistry background with SV University, Tirupati. And then 77, I got again moved to SV University in botany with applied plant physiology as the main specialization. And then I joined as a research scholar and then joined as a curator of herbarium in 79. So that it took a while for me to get a PhD. So 1984, I got PhD in botany with applied plant physiology. So 1988, I was awarded by the government of India to go on the study abroad program. So I came here. One of my relative was here, so that's the reason I came to Mississippi State. Otherwise, I would have gone to some other place. So I was one of the two students, two visiting scientists selected by government of India to send for study abroad program. So I came to here, and then I landed at Mississippi State. A year after that, then we started. They offered me the job, so I stayed in the same place, same room, almost like 37 years. So today I'm going to talk about 4 different areas of research. One is looking at the trends, signs, signatures of food production systems of the past and also on the future, looking at what is ahead of it. And then I'm also going to address a little bit on the facilities, what kind of facilities we need to address climate change related issues, and also the tools in the terms. Tools in my sense is crop simulation models kind of things to address climate change and crop responses to climate change. And then the other topic is mostly I concentrate on climate change and crops. Climate stress is mostly on crops, and then with some conclusions. So as we all know that this is not anything new, but the population is on the rise. Most recently, it's increasing exponentially. As of today, we have about 8.2 billion people. By 2050, which is in another 25 years, we are going to expect around 9.8. That is only in 25 years, which is not too far away. And then we are adding almost like the size of Indian population or more in the next 25 years. By 2100, we are expected to reach around 11.2 billions. If you look at the plan, if you look at what we need to address is plants, they produce about 80% of the food that we eat. So protecting their health and also making everything happen in a sustainable manner is the critical thing. So if we look at one of the issues that we are using is in the next 25 years, the current yields, some people are already hungry, but we are in the next 25 years, we are expected to reach, add almost like 1.6 billion people. That is almost like combined, almost very close to combining India and Pakistan population together. That many people in the next 25 years. So if you look at these four of the crops, they provide about 80% of the calories that we eat, more or less. 4 of the crops: corn, the maize or corn, rice, wheat, and soybean. If you look at the last 60 years, the yield has increased. We had to pat ourselves that it increased tremendously at the rate of 1.2% per year. But we need about 2.4% increase per year to meet the 2050 population, the population that is expected to be in 2050. And the population is also going to be with a higher earning capacities. There are also going to be some sort of diet shifts. And also there is some competition coming from increasing biofuel consumption as well. Can we do that 1.2 that we haven't achieved over the last 60 years in the next 25 years? That's a big challenge. It is by 2050, I may not be there, but many of you, the young people, are there to make that one happen. So there is another challenge that also looks at it is because we have as we move along these years, the population is going to have a little bit more affordability. They will have more an affluent population. Their food per capita consumption will be all the products, like if you look at vegetables, or selected fruits, meat and poultry, flowers and cereals. They're all expected, the consumption is going to increase per capita basis. The reason for that one is they can afford it more, they can buy it, and not that many people going hungry to a certain extent. So as a crop scientist, we have to worry about this one also, not only the population, but this consumption rates of per capita consumption. There is another stats that also makes some sense in that. You look at the right hand side is most of them related to animal system. Why do you worry about animal systems? Because there is about 24% of the food that we produce is going to for animal feed lots and particularly to animal production systems. So if you look at the poultry here on the bottom, next one is the pig or the pig meat here, and then beef and buffaloes, and then other meats are there. So when people are having a few a little bit more comfortable with the money, so they demand animal-based diets. So that's the reason we are consuming that consumption of those, our production or consumption are going up for the food system. As an agriculture scientist, we need to worry about those also, like that diet shifts. That's what it is. The more affluent population will demand meat-based diets. So that's one of the other thing also we have to worry about it. The other big challenge is about the water. Water, it is not only the crops are thirsty, and also the water consumption is increasing, and majority of the fresh water, that is 70% of it, is being used for agriculture. Today we are consuming about 4.3 trillion cubic meters on an annual basis. 70% of it is going to the agriculture, and then about 30% goes to industry, and then another 10% goes for other aspects. So if you look at the right hand side, then this is liters of water to produce 1 kg of product, and listed on this right hand side here. Meat is this one, 15,000 liters for 1 kg of meat. That mostly beef, nuts, and then sheep and goat meat, pig meat, chicken, and then the list goes on. The bottom one is the vegetables and fruits are at the bottom hand side. So if there is some sort of issues, we are already seeing it between countries, or if the rivers are flowing between countries, upper catchment area, lower catchment area, delta areas, conflicts are getting already in place. Within the country also they're getting conflicts because they're not releasing water to one state to the other states, kind of thing. This is a big issue. Water is going to be a big issue. Globally, freshwater consumption is increasing because increasing population. So John F. Kennedy, former president of the United States, and one time he told that anyone who can solve the problem of water will be worthy of 2 Nobel Prizes, one for peace, and the other one for the science itself. That is true even today. You can see that one that is happening between countries when the rivers move or flow through the different countries, or within the country also. Some flows through different states, each state demanding more or less, kind of thing. Therefore, water fights are already in place. There is another statistics we have to worry about it. In this one, I have listed on the right hand side is regions. Next one column is the cropland area. The next one is the irrigated area, million hectares, and then salinized area, the last one. If you only pay attention to this in parentheses, which is blue text, that is the percent change from 1985 to 2000. So if we do not use a sustainable irrigation practices, the difference between this third column to fourth column is irrigated area. 22% irrigated area contribute 14% in China. In India, the irrigated area is about 31%, contributes about 50% of salinized area because we are not using sustainable irrigation practices. Globally, there is about 21% irrigated area, contributes about 37% salinized area. So this is also an issue because we don't have a whole lot of land to bring in. So we are taking, if we don't use the sustainable irrigation practices, our prime land is going to be unsustainable. They're not be that productive because of this increased salinity and unsustainable irrigation practices to a certain extent. And the other one is a lot of people worry about the connection between climate change, changes in temperature or changes in climate to changes in greenhouse gases. But if you look at temperature, for example, no two seasons are equal. Here I'm showing where I'm showing this seasonal temperatures changes for 2 different years compared to the average temperatures. If you look at the red line, which is 2011, and the blue line is 2001, those two are different. That's what they are projecting to change in climate as well. And then this is the Mississippi average temperatures. A long-term average is the black line, and then a red and blue line are 2 different years, just to show that differences are there. The seasons are different. Each season has its own temperature variability in the temperature as well. On the top of these, all these trends that we have witnessed, we are getting that's what we call it as a global climate change. This is one of the challenges that we are going to face it. The greenhouse gases are increasing, that is monitored by sensors, so there's no doubt about those things. So we are changing the chemistry of the atmosphere. All these greenhouse gases that include CO2, methane, nitrous oxide, etc., they're all increasing. They're being monitored across the globe, several different locations that keep increasing over the years. And then if you do the same thing, they are expected to increase in the future as well. With the changes in chemistry, the temperatures are projected to increase, and also the asymmetry in temperatures are also projected to increase. The asymmetry in the sense that the difference between day and night are shrinking, so the nighttime temperatures are projected to increase a lot more in the future than in the daytime temperatures. So the difference between day and nighttime temperature getting closer and closer and closer as we move along with the changes in greenhouse gases in the future. Along with these greenhouse gases, glaciers are getting less water, less, and then oceans and sea levels are also slowly rising. As far as the precipitation patterns, they are kind of spatial variability is there. So some regions will get more, some regions will get less. So the drought intensity is irrespective of the region. We can expect more intensities are going to come more frequently, more intense in the future conditions. The other one is changes in the ultraviolet radiation due to the depletion of ozone levels. But there is some kind of change that implemented by the International Agency for what we call it as a Montreal Protocol. They're phasing out the ozone depleting chemicals. So the ozone levels are rebuilding to a certain extent, but other chemicals are increasing because of the replacement of aerosol content, that is refrigerators and other things are being, some other chemicals we are putting in the atmosphere. We really don't know what the long-term consequences of those chemicals. With all these, then there is a frequency of extreme events, either in terms of high temperature followed by drought, extreme drought condition. Those frequencies are projected to increase in the future. If you look at only the CO2, as far as the CO2 is concerned, it's good for plants because that's a raw material for plants, and it is a raw material for photosynthesis. So it's not a bad thing for plants. Plants, either it's crop plants or natural ecosystem, it's good for majority of the plants because it's a raw material. I'll show you in the next couple of slides. These are the measured CO2 concentration at Mauna Loa, Hawaii. It's been the same trends are being observed in several different locations. And these are the ice core data. They drill a core into the ice core, and then they analyze the gas trapped in the ice at different ages, and then from that they reconstructed the CO2 concentration over many previous years. So that goes hand in hand. So the rise in CO2 concentration is due to carbon emission, either due to burning of fossil fuels or some due to natural causes also. So as far as the plants are concerned, raising CO2 is not a really bad thing as far as the plants are concerned, but the indirect effect or the direct effect of changes in greenhouse gases and other things on other climate issues are the concern. One of them is changes in temperature. If you look at the long-term global temperatures, most recent years, you can see that most of them are increasing. Average temperature, surface air temperature in July, for example, this one is hottest years or most recent ones. So it is over the years, it is increasing. Global temperature to certain extent over the past century, there is almost close to 1 degree increase, almost like a 1 degree increase. There, if we do the same thing what we are doing now, we can expect changes in global temperature is going to continue. And then there are different kind of climate modelers. They predict different scenarios. What they call it as a representative concentration pathways, they call it different. So the minimum is anywhere from CO2 equivalent is anywhere from 450. We are already at 425, but 450 to 1250 ppm CO2 equivalent greenhouse gases by 2100. With that, changes in greenhouse gases, the climate models predict the global temperatures will go up to 1.5 to 6 degrees centigrade. Then you can imagine if it really goes anywhere, even if they close by for about 4 degrees and the middle point kind of things, even that one is really extreme. For some of them, like if summertime or late March, early March in Andhra Pradesh, if we add like 4 or 5 degrees to that the current temperatures, you can imagine what the intensity of those temperatures on human and animal health, and also plants where those are not sessile because they cannot move, so they will be suffered a lot more than the human systems. So the other one is something like what we call it as not only global warming or climate change, but also they are expected to percent change in nighttime temperature anywhere from 40 something like at 18% or 20% to 40% in the next 75 years. So they're getting day and nights are going to get nighttime temperature are going to go inches closer to the daytime temperatures. That's what they're projecting to. That is also a concern. Why are we worried about too much about nighttime temperature? That also, when nighttime temperature goes up, typically if it goes beyond the optimum temperature for any crop, then it burns a lot more carbohydrates. That will decrease the yield to a certain extent. That's the reason we are concerned about nighttime temperature, changes in nighttime temperature. So here I'm showing to show you different regions. This one is average temperature for Mississippi, the bottom line. This one, and then this one is a blue line is Phoenix, Arizona, which is a desert area in the southwest of United States. And then the green line is, this one is given to one of my postdocs, is Hyderabad, India. and then the monsoon season starts, and then it drops down a little bit, and then that's what it is. The other one is red line is Maros, Indonesia, which is 5 degrees south of equator, one of the island nation, very close to the equator. So the one thing I wanted to point out is that the seasonality is very less and closer to the equator. Closer to the equator, there is not that much seasonality. So if there is a change in temperature, these temperatures will be above that, whatever the above throughout the year, it will be about certain numbers. But if Mississippi temperature gets warmer and then it becomes the blue one, instead of planting cotton or some summer crops here, they can plant here a little earlier. They have that planting adaptations. But if it is closer to equatorial region, they do not have that one. This line goes above that one. This is a 15 degrees mark, which is a minimum temperature for many summer grown crops. And then this is about a 28 degrees mark, which is optimal temperature for many fruit crops, even including cotton and other rice. There is some variation about the optimal temperature, but that's what for cotton, that's the optimal temperature for reproduction. So the point I wanted to make is regions or countries closer to the equator, they have a less adoption to use it. It's like because if this one goes, it goes above that line all through the year because of less seasonality. So any changes in the environment that will have an impact on the food production systems. So over the years, I have been working on several of the environmental stresses. I worked on almost like a 20 different crop species that include some of them are horticulture crops as well. I probably have almost like as many as almost like 30, 40 publications in horticulture crops. But today I'm going to show a few examples from each one of them. CO2, as I said, it's a raw material for plants for photosynthesis, so that's good for plants as far as we know. There are some issues related to that one on food production system, but as far as the plants are concerned, that increasing CO2 is good for it. Any changes in temperature along with the changes in CO2 will, including extreme temperatures, I'm going to be talking about a little bit on the temperature effects, and then solar radiation, including UV radiation on crops, and then precipitation patterns, drought intensities, flooding. These are all these factors that affect plants. Flooding, more recently, we are because of the intensity of rainfall, flooding is become like an increasing problem in our area also. Early season flooding is kind of becoming like a problem. We cannot plant because it has a lot of rainfall and the soils get too much saturated. Soil minerals, including heavy metals, salinities, another issues. These are the factors that affect plants. So I will be talking about a little bit of some of those effects on crop plants, giving some examples. Here I wanted to show you one of the other important thing, even though this is the data is from the US. These are the crops that are listed here. Corn to wheat, soybean, sorghum, oat, barley, potato, sugar beet. And this is the highest yield recorded for that crop. Highest yield kgs per hectare. Highest yield. This is an average yield. Average yield for that same crop in the US. And then they also listed losses due to diseases, insects, and weeds. And then if you look at the physiochemical, that's the last column here. That is the highest yield recorded this yield minus average yield plus disease plus insects plus weeds. That is what we call it as a physiochemical. That means environmental stresses. On average, there is about 67% loss of the potential. Means they have the recorded yield. These are actual recorded yields. Somebody actually produced that one. There has only 4,600 kgs per hectare corn yield on average, but 19,300 is being produced. Some of the farmers produce that one. So what I'm trying to say is that there is about 67% loss is due to environmental stresses that I listed in the previous slide. So, so what are the tools or facilities needed to study environmental stresses? One of them here in the US, mostly these are mostly done, facilities listed here is open top chambers. Means they can enrich CO2 concentration in these chambers, and then the other one, and then other factors are not controlled. There is some sort of um, walls, chamber walls kind of thing. Some issues are there. Only CO2 they can control, but they need a lot of CO2 to inject into to maintain certain levels of CO2. Currently, the CO2 concentration in the atmosphere is about 425, close to 425. In 1950, it was 315 ppm, 315 ppm, parts per million. Today, 425. It's that much increase. And then there is another facility that in Phoenix, Arizona is what they call it as a free air carbon dioxide enrichment experiment. They call it as a phase experiment, free air carbon dioxide enrichment experiment. So they have the vents, and then they open the vents based on the wind direction and wind speed. They open the vent to enrich CO2 inside those rings. So this facility provides a little bit additional space, and they also added some sort of infrared heaters to those phase experiment. They call it the T-phase experiment. These are some of the facilities that, particularly looking at CO2 and temperature interaction on crops, these are some of the facilities. The other one is open top chambers. Then they have something like a indoor plant growth chambers, some sort of hoop houses or greenhouses. Some of these we do it for ourselves, creating our own small greenhouses kind of thing. We move those tents into this, onto this. When we want to monitor some growth kind of thing, you just move outside. When you don't need it, we can discard that one. So this is a kind of, these are, hello?
No cross talking, please. Sir, proceed.
Okay. These are some of the facilities. These are the greenhouses that we have recently constructed. One of them, this one is about $1.5 million. So this is just one greenhouse. So these facilities are expensive. On the top of it, this what we call it, this is my lab, Environmental Plant Physiology Laboratory at Mississippi State. It has what we call it the sunlit plant growth chamber facility. These are kind of a small, not really huge chambers. This is about 2.5 meters tall, 1.5 meters wide, 1 meter soil bin is there, and more or less they're sunlit chambers, means the plexiglass allows 95% of the sunlight passes through the plexiglass, and then the plants are grown almost in the natural sunlight level. That's the reason they call it as a sunlit chambers. The problem with this one is you have a limited space, but most everything you can control it inside it under natural sunlit conditions, no artificial light. The tools that I was talking about is a kind of thing. We developed a cotton model that is being used by the farmers and also the policy and the researcher to predict. Cotton is produced in the 14 southern states in the United States because based on the temperature conditions, these are the 14 southern states, they produce cotton. And this is what the each pixel level yield levels are in those 14 southern states. This is simulated yield using this cotton model. It's a mechanistic model. So if the model is mechanistic, and then if you can simulate very close to what is observed values, then you can use it for management. You can use it for policy, assisting the policymakers in decision making things. As I said, the next few slides, I'm going to talk about these two main processes. One is photosynthesis. This one is taking CO2 inside the leaf. The other one is water loss coming out of these leaves. So if you take something like, you all know that one, but that's if you take a pen and put a dot on this leaf, this is a cotton leaf, for example, it has about 250 stomates per 1 square millimeter. That's one dot will have that many stomates. That's so the plants, they take 6 molecules of CO2, and that everybody, we all know in the plant science, and combines with water and converts sun energy using the plant systems, 1 molecule of carbohydrate and releases 6 molecules of oxygen. This is the basic mother process of photosynthesis. So as far as we know, raising CO2 concentration is good for plants because you can produce more carbohydrate. That's not a bad thing. So first I'm going to show few of these CO2 intake which is related to photosynthesis. Then I took a couple of examples of water loss from the plants. When the stomates are open, there's a physical process, physical concentration gradient difference. Outside is more, inside the leaf is less. So the physically CO2 moves inside the plant, and then it gets fixed and then transported to the different plant parts. The same way goes with the water loss also. Inside it's very moist levels, unless it is very dry leaf. So outside is very dry, so it physically escapes from this. When the stomates are open, they physically escape. Those processes are gradient different processes. I wanted to give you a little bit of evolution and ecology in this picture to understand what's happening. When land plants evolved about 500 billion years ago, 500 million years ago, the atmosphere has only CO2 in the atmosphere, no oxygen at that point. So at that point, the plants, they developed what we call it as the ancient photosynthesis system, what we call it as C3 photosynthesis process. That enzyme that facilitates that C3 fixation of C3 photosynthesis is RUBP carboxylase. At that point of time, maybe that is a design flaw in the evolution, but instead of getting a C4 photosynthesis at that time, they got it a C3 photosynthesis. That enzyme is not specific to CO2 only. It bombards with oxygen, it goes into oxygenation process, and then there is a loss of energy in that system, photorespiration they call it, and then if it bombards with the CO2, it goes into the carbohydrate production. So that's a design flaw in the evolution to a certain extent, because if they had developed a C4 photosynthesis to start with, we would have a C4 forest, but we don't have it now. If you look at it, there is about 250,000 higher plants are there. Higher plant means flowering plants, dicots and monocots. 89% of them are C3s, mostly trees and others, shrubs, all sorts of things are there. Majority of all the trees are C4, except 1 or 2 small shrubs or medium-sized C4 plants are there, but that's also very rare. 1 or 2 species are there. About 3.2% of them, or 8,100 plants, are C4 plants. The reason I'm giving this one is the other one is a CAM plants, which is also another form of C4. That's 8%, which is a CAM. One of the horticulture crops is pineapple. Pineapple is a CAM plant. One of the C4 dicot plant that comes to mind is a grain amaranth, or leafy amaranth, that we use it for curry, making papu or something like in Andhra Pradesh kind of thing. That's one of them. So when about between 25 to 32 million years ago, due to convergent evolution that led to the emergence of C4 plants, what they have done is they pushed that inefficient C3 photosynthesis into this bundle sheath cells, and they created a craniotomy, put the whole C3 photosynthesis inside this, and then they recreated another chemistry that fixes carbon with PEP carboxylase enzyme into a C4 acid, and then that C4 acid is transported into this bundle sheath cells. The bundle sheath cells are very thick, and then the CO2 C4 acid is decarboxylated and CO2 released, and that is that the ancient photosynthesis is supersaturated with with the CO2 inside those bundle sheet cells. For that reason, C4 plants are much more efficient under lower levels of CO2 concentration than majority of the C3 plants. So C4 plants, they appeared in 25 to 32 million years ago due to this, what we call it as a convergent evolution, means looking for different species, looking for the same kind of adaptation, something like having bats, birds, and insects having wings are the common thing to fly, something similar to that one. There is about 8,100 species are so far identified, and they represent about 3.2% of the higher plant species, and they contribute about 23% of the terrestrial carbon fixation now. The problem of what I wanted to bring it to that one is that this is a response to CO2 concentration in the x-axis, and then photosynthetic response here. This is the C4 photosynthesis response. Anything less than 420 ppm, that is today's concentration. The C4 plants are much more efficient compared to the C3 because of that ancient photosynthesis that is there in the C3. It has that inefficient enzyme that acts like based on the CO2 concentration and oxygen concentration in the leaf inside the leaf. There is a kind of waste and wastage of the wastage of the process in terms of what we call it as a photorespiration in C3 plants. When it is freely available, in the atmosphere due to increase in CO2 concentration, the C4 plants have to do that chemistry, fixing here and then transporting here, retransporting the substrate between these cells. This is a mesophyll cell, this is a bundle sheet cell. They have to fix it in these cells, transport it into this one, and then the substrate has to be transported back into the mesophyll cells. When it is freely available, they have to do that chemistry that involves some energy, and energy is involved. When freely available, it goes to that crossover CO2 concentration is around 420 ppm, 420, 425 ppm parts per million CO2 concentration. So we have to see whether that much corn is going to be grown in the future. Corn is a C4 grass. Whether it will be grown in the future that many acres as they are grown as of today. That's corn is one of the largest cropping system in the world currently. It superseded even wheat and other crops also in terms of hectares that are being grown across the globe. So what we are making, the point I wanted to make is C4 plants, evolutionary, it took, the first thing that happened is the movement of plant system, that is C3 photosynthesis. The second system, it took almost like 25 to 32 million years back that happened. Almost like 500 to 25 million is a lot of millions of years are there to get something, a C4 system. In the matter of few years, we are making those C4 plants a little bit irrelevant because of increase of polluting the atmosphere, and then the C4 plants are going to get a little bit more advantages. That you may think that it may be good for majority of the wheat is a C3 crop, rice is a C3. That may be good for those crops to a certain extent, but the thing is there, the other things are also there along with the C4 photosynthesis. Here has come a couple of examples. This one is amaranthus, which is a leafy green or green amaranth, that's called. There is no response to CO2 concentration. Either 420 is almost like today's concentration, 100 ppm less than today, about 820 ppm at the right side of the cotton, responding a little bit favorably, which is a C3. C4 sorghum is not that much response. C3 soybean, a little bit increase. C3 wheat, little bit increase. but C4 plants are not responding to elevated CO2. So changes in CO2 may not be good for C4 plant to beyond what we have currently in the atmosphere. If you look at C3 plants response to CO2, you have to look at currently we are here. Before the current, below that current level, there's a kind of linear response. Beyond the current level, the response is not linear. There's a small increase with increase in CO2. So other factors are come and play. So there is about a 18% increase in photosynthesis between 1958 to 22. The CO2 concentration 1958 is about 315, 22 is about 417, but that's about 18% increase in photosynthesis. Just increase in CO2 concentration contributed about 18% increase in photosynthesis in cotton. That means raising CO2 concentration contributed about 18% of the yields as well between those two times, 1958 to 22. And also I wanted to point out that one is here we are looking at solar radiation. This is a sunny day, very clear day. You get this much light. If you get a partial cloudy day, you get this much light. If you get a very cloudy day, you get this much light. This is the total megajoules per meter square per day. Here we are looking at a whole plant photosynthesis, grams of CO2 per meter square per day. These are plants grown at elevated CO2, plants grown at ambient CO2. You can see that the plants are getting benefited with increase in CO2 at a wide range of solar radiation levels. This is a red one is the elevated CO2 that is around 720 ppm compared to 360 ppm. Then when this experiment was conducted, when I came around early 90s, we were conducting at 360 ppm. That was the outside ambient CO2 concentration. Currently, we are conducting around 425 because that much increase in CO2 concentration. The point I wanted to make is, in the future, plants grown at elevated CO2 will get benefited at a wide range of radiation loads. Either cloudy day or sunny day, they will get that benefit is going to be there, particularly for C3 plants. And looking at C3 plants again across a wide range of temperatures, this is a little bit low temperature. These two are moderate temperatures. These are high temperature on this side. Relative to Mississippi, this is ambient CO2, elevated CO2. Ambient is at the bottom, elevated is on the top. You can see that cotton responds very nicely to elevated CO2 concentration for the whole growing season. Looking at canopy photosynthesis, milligram CO2 per meter square per second, time after planting, you can see that whole season it's getting benefited with increase in CO2. They have additional benefit at a wide range of temperature conditions. The other factor that impacts plants is photosynthesis, particularly looking at photosynthesis, is leaf water status. Leaf water status means based on the environmental demand and root zones, soil moisture content, you can determine the leaf water potential. These are well-watered plants. When you give all the water they needed, they show when you go and monitor at the noontime, they will have this kind of values, minus 1.0 to 1.5. These are well-watered plants. Will show. These are mildly stressed plants. These are severely stressed plants. The blue ones are ambient CO2, red ones are elevated CO2. The 2 points I wanted to make it. Photosynthesis, which is almost translates to biomass production. These are canopy level photosynthesis, translates to biomass production or ultimately leads to yield. They are declining irrespective of CO2 levels. But at both CO2 levels, they are declining linearly with increase in water stress. That's the first point. The decline, the beneficial effect of increased CO2 is there across a wide range of soil leaf water potential, means under wide range of environmental drought stress conditions. So that is also there. Plants grown under wide range of drought conditions in the future. So if CO2 goes to 700, that benefit is there across a wide range of drought condition compared to today's or 360 CO2 level, 350 in this case when that particular experiment was conducted. The other one is ultraviolet radiation. So many people may not, if you look at, if you go and expose to yourself to sun, outside sun levels, you work outside, your skin get a little bit tan color because of the increased UV radiation that you expose to higher UV radiation. So that you will have that one. So the current UV levels are anywhere here around 5 to 6. At Mississippi, it varies depending upon the location and also the height of the altitude of the place. So current levels are somewhere here. So if you look at these are ambient CO2 levels, current levels, red ones, looking at photosynthesis on the y-axis. This is in the 720 CO2 levels, that is expected around 22,100, 2050 to 2100. So the plants are getting benefited with increase in CO2. They are going to get little benefit. Elevated CO2 is going to be beneficial across a wide range of UV levels. We have seen wide range of temperatures, wide range of wide range of sun levels, and then wide range of water conditions, wide range of UV radiations. Plants are going to get beneficial effect with additional CO2 added into the atmosphere, particularly for C3 plants. The other aspect is nitrogen. Nitrogen has an effect on photosynthesis as well. These are plants with well fertilized cotton plants. They will show anywhere from 4 to 4.5 leaf nitrogen concentration. These are mildly stressed plants for nitrogen. These are very stressed for nitrogen, lack of nitrogen in the soil. So there is a linear response from here to here, and then beyond that, there's not that much response. The 2 points again, similar to the other responses, both either grown today or in the future, the response is similar, almost like parallel response. And plants grown at future CO2 are going to get additional benefit across a wide range of nitrogen levels. That's another point I wanted to make it. So we looked at, as far as the photosynthesis is concerned, for C3 plants, the increase in CO2 concentration is going to be beneficial across a wide range of stressful environmental conditions for C3 plants. For C4 plant, that benefit is not there. That I will show you in the next slide. The other one I wanted to talk a little bit on that water loss from these plants is if you look at C3 plants like cotton, 89% of the higher plants are similar to this response. We're going to be showing similar to cotton response, which is cotton is a C3 plant. So plants gone at ambient CO2, if they're losing around, based on the temperature conditions, about 250, 250, elevated CO2, they lose about 150 milligrams of CO2 if we are monitoring at a leaf level basis. But if you are monitoring at a canopy level basis, they're not that much different. The reason for that one is plants grown at elevated CO2, they produce additional photosynthesis that will increase the number of leaves, increase the leaf area to a certain extent. Instead of having plants grown at ambient CO2 here, elevated CO2 here, leaf area, meter square per meter, meter square per plant. If this one is, there is about 13% increase in leaf area. That increase in leaf area upset the leaf level loss. The net result in cotton, at least in cotton, there's no additional advantage for water loss is not there. So that's plants are going to be losing almost the same amount either today or in the future. Similarly, for C4 plant, as far as I said, this is C4 plants, because they have to fix and transport and retransport the substrate between two different tissue systems. They're spending that energy, so they're the CO2 response to elevated CO2 response is not there. This is a net photosynthesis for sorghum, which is a C4 plant. At ambient CO2 levels, net photosynthesis are elevated CO2, that response is linear, and then they go hand in hand. There is no response to elevated CO2, as I said, because the crossover response is beyond 420 ppm, so there is not advantage for C4 plants. There is about 5% decrease in transpiration rate in sorghum. Looking at the canopy level transpiration, there is about 5% decrease. That may be a little advantageous because they can survive and a little bit drought conditions much better in the future. If CO2 goes up, C3 plants are going to be doing a little better under future CO2 conditions. The other point, what I showed is the main photosynthesis process that will translate to the biomass production. But if you look at the temperature effects on total biomass production, straw weight, here looking at cotton, rice plants, straw weight means without the grain weight, above ground straw weight, without the grain. I'm here, two varieties are there. Tropical is Rex is the variety that we grow in the here at Mississippi, and tropical IR6 is the variety that is grown in Andhra and other Asian countries. The temperature response is not that much there for the whole season straw weight. If you look at the grain weight, the response is much different. Straw weight is here, not much response to changes in temperature is not going to make that much difference. So across the board, there is some sort of small variation is there, but not much. But in terms of grain weight, that is much different. This is the grain weight grams per plant. The optimal temperature for Rex is somewhere here. For IR6 is somewhere here, which is around 26 degrees. This one is around 24 degrees. And then the decrease from their respective optimum temperature, there is about 10% decrease for each increase in temperature from the respective optima. For this tropical, there is about from here to here, there is about 10% decrease. So any changes in temperature, 1 degree change, there is about 10% decrease in yield for those crops, either tropical temperate or tropical rice varieties from their respective optimal temperatures. That is one of them. Other one is there is some concern about the nighttime temperature, rise in nighttime temperature without changing the daytime temperature. This is the control. This is the 10% increase in nighttime temperature with the daytime constant to that one control, and then 20% increase in nighttime temperature with the daytime constant temperatures. We see some sort of sterility, like a white without filled grains, blanking. You can see one here, one here, one here, one here, one here, and then one of them here with a 10% increase. In the control, we do not see those things happening. So that something that is disrupting this grain filling process or the fertility process. If the nighttime temperature goes up, particularly in the rice. Same thing happens for sorghum as well. Sorghum is another crop that is dryland crop or even irrigated to certain extent. There are one tolerant variety, one sensitive variety. Again, there is about change in nighttime temperature also from the respective optimal temperature. Again, there is almost a similar decline is there. Sensitive variety, the decline is a lot more than the tolerant variety. There is some sort of varietal variation is there in sorghum to a certain extent. Again, the increase in temperature is going to have some effect on sorghum yield as well. Cotton is another one. Vegetative growth is not that sensitive to change variability in temperature. Even 40, 32 day-night temperature compared to these are 4-week-old cotton seedlings at different temperature conditions. You can see that optimum growth is somewhere here, and the number of leaves are more at this high temperature compared to the low temperatures. But you can see that temperature doesn't have that much influence on vegetative growth. And if that is reflected here, like we have a wide range of temperatures, 1995 ambient temperature -2 to 95, Mississippi temperature +2 degrees, +5 degrees, +7 degrees. Plants gone at ambient and elevated CO2 with the green. You can see that C3 plant, cotton being a C3 plant, it has an added advantage in producing more biomass at a wide range of temperature conditions. But the temperature effect is not that much across temperature conditions on vegetative biomass. But if fruiting sites, number of fruiting sites produced, it's not a problem. Fruit retention is the problem. That is related to at high temperatures. The pollen that is produced is kind of ill-formed and they are not fertile. They won't fertilize the fruit. So that's the problem. Like pollen sterility issues are the issues that are related to high temperature that is not getting any beneficial effect. For cotton, that optimal temperature for reproductive growth is around 27 degrees, 28 degrees. And then there is about, again, if you put a regression equation, there is about 10% decrease in yield from the respective optima with a 1 degree increase in temperature again. So high temperatures mostly related to pollen fertilization, pollen quality of the pollen. So those are easy to study, and anybody can do it in using a microscope and a small incubator kind of things. You can do that kind of things. Even electron microscopic, scanning electron microscopy can do that pollen image analysis as well. Anybody can do that kind of thing. So here is another example for soybean. Across a wide range of temperatures, the optimum temperature for soybean is around tolerant variety is about 25 degrees. The less tolerant variety is about 24 degrees, not much different, but the decrease, the tolerant variety is 10%. The less tolerant variety is about 23% for each degree increase in temperature. So it's again, it is following the similar trends like the other crop species. This is on the drought, well watered here and less well watered on this side. This is the total weight of the plant. This is the seed weight of the soybean plant, and then harvest index almost declines. Means harvest index is almost like a reproductive biomass divided by the total biomass. That gives how much is partitioned to the grain weight kind of thing. Here you can see that 100% water and then 20% of under drought conditions here on the right hand side. You can see the number of grains, size of the grain, and the total number of grains per plant also declined. 10% of the seed is shown for each plant. In this image, there is about 10% of the seed per plant are shown instead of putting the whole plant weight. So you can see that one is declining with increase in decrease in moisture content. There is another issue that is related to flooding is what happens in the flooding conditions. So there are a couple of things happens in the flooding. So that when the crops are flooded, if there is a standing water in the cropping systems, the soil oxygen concentration declines very fast. It goes from here within 5 days to almost like 18-20% in the soil oxygen. It goes to 0% in the soil oxygen content within 5 days. So that's what we call it as a hypoxic conditions. And then it goes to the anoxic conditions. As long as it is flooded, it goes to anoxic conditions. So instead of during this process, there are a lot of things happens in the plant, particularly in the roots. Instead of aerobic respiration, they go to anaerobic respiration. The number of ATP molecules produced under aerobic respiration is much different than anaerobic conditions. So that will affect lots of other things apart from the growth and developmental aspect. You can look at here, control no flooded condition, 2 days of flooding, and then 14 days of flooding here. Like you can see the decline 2, 4, 6, 8, 10 days of flooding and taken, the picture was taken after 14 days. So that many days of flooding, you can see that even 2 days, 3 days of flooding has impact both on root systems and also the shoot systems, and that also happens on the physiology that will translate into those things as well. Other aspect is temperature. Another one is here is the corn, the water, drought conditions, lack of water. You can see the decline in biomass. Lack of flooding, you can see the decline in biomass, and also intensity of drought stress during flowering and then grain filling stages. You have some effect, differential effect. Flowering has a lot more effect than the grain filling stages. That also some imbalance in growth of um, the kernels and the size of the kernels and the size of the cob in corn. And the other one is the temperature effects, and also this one is the nitrogen effects. Less nitrogen on this side, more nitrogen, optimum nitrogen on this side. You can see that those environmental stresses have different differential effects on different processes. So that will have an effect on it. The other component that many people do not have that much idea of how the ultraviolet radiation coming from the sun is going to have an effect on plants. We are one of the labs that we have been doing this UV radiation effects on plants because our facility can induce or put UV lamps inside the facility, inside the chambers, and then study their effects on plants. So what I said is about average ambient ambient UV levels are somewhere about 4 to 5 kilojoules. That's what it is. So if you have more than that one, certain regions have up to 9 to 10 kilojoules. That will have an effect on one is 100%. Anything less than 1, like a 0.8 is about a 20% reduction. 0.4 is about this much reduction if we have 15 kilojoules. Some regions in the US will get around 10 kilojoules on certain days. So that will have an effect on that one. So it's a kind of, may not be able to notice that one, but you can see that the ambient UV level, certain regions, it gets about 10 kilojoules in the summertime, in the middle of the summer, compared to the early in the season. So it follows the total solar radiation in the day throughout the growing period. That follows almost mimics the total solar radiation. So that will have an impact as well on many crops. There is another one is the rangeland, native grassland. Mostly it's anything that related to rangeland grasses. This is the C4 grass. As I said, the C4 grass, the response to CO2 is not very small. This is the ambient, this is elevated CO2 across a wide range of temperature for vegetative biomass. This is for the reproductive biomass. So the responses are very small compared to the C3 crops that I have shown in the previous slides. Vegetative biomass is decline in vegetative biomass is much less in C4 grasses at a higher temperature compared to the decline in biomass and reproductive mass. So in other words, if our area, particularly in southern Texas, where it is very warmer, if they go out of cultivation for cotton, you can put this kind of rangeland grass and still can grow and produce grass that can be fed to the cows and animal systems. That is an option. So shifting cropping systems is another option that with the change in climate also, that's an added advantage. The other one, we did some work on what we call it as horticulture crops. Some of them is sweet potatoes. Sweet potato is one of the Mississippi is the second largest sweet potato producer in the country. Here in the US, we grow orange flesh sweet potatoes in the US. It has more vitamin vitamin A compared to the white sweet potatoes that were mostly grown in India to a certain extent. This is the temperature response. This is the CO2 response here, ambient and elevated. You can see that one is getting beneficial effect because sweet potato is a C3 crop. It has a beneficial effect. The one thing I wanted to point out is There is a added advantage. The roots are larger if the temperatures are anywhere 35, 27 and below. If it goes beyond that temperatures, either in the future or in the present condition, the storage roots are not enlarging it. So the high temperatures are causing some effect on enlargement process of the storage roots. So the bulking process is being affected. Then you get some sort of a pencil-like root systems, that is, those are not edible, and if they are like this. So that almost mimics the grain kind of conditions in other crops. This, even though it's a vegetative structure, still it mimics almost similar to that one. So increase in temperature is not that much beneficial, even for this sweet potato production. And increased CO2 is not getting any beneficial effects at high CO2 conditions, even for the vegetative, the storage roots, what we call it as what we buy in the stores, what we call them as the storage roots or sweet potatoes that we buy in the market. So elevated CO2 is not getting any beneficial effect at the high CO2 conditions. We also did some on the kale and mustard, some of the leafy vegetables. These are the control, and then this is a plus UV and plus high temperature, plus CO2, plus UVB CO2, plus T temperature, plus CO2, and the combination of these things. This is the plus increase temperature, increase CO2, increase UV radiation. If any kind of stress conditions are there, the biomass production is harvestable biomass is reduced. Increased CO2, this is also kale or mustard. Those are C3 crops. It's always beneficial for those things. So you can get them, you can produce more harvestable biomass, means and the heads are a little larger in size and they weigh a little bit more. Quality wise, there may be small changes in quality differences. That those differences may be a little bit minor for certain crops, but if you look at quality aspect, there are few things. This is the grain changes in yield with the temperature, with CO2. There is some sort of that much increase in yield, and change in total weight is also this one, but it changes in yield that much. With the CO temperature, there is a decline. With the drought, there is further decline. But if you look at in terms of changes in concentration of starch, increased CO2 has an additional starch is produced, particularly in C3 crops. Temperature has a little negative side, but increased CO2 that has more starchy content in the grains and also in the leaves and stems as well. Protein content, increased plants grown at elevated CO2 has a less protein. That has some sort of implications. In other words, if you want to use the same amount of protein, you need to use a lot more material. Particularly, that will upset a lot of insect biology, or the human consumption also. I guess you have to eat a lot more to get the same amount of protein if the plants are grown at elevated CO2 compared to the ambient CO2, because of the protein concentration of the protein is much lower for plants grown at elevated CO2. The other component that is of concern is these gliadins and glutens. These are kind of grain quality issues. If they are kind of decreased with elevated CO2, then there is a problem with that one, because the reason for that one is they help the bread quality to a certain extent. If those things are affected, then the quality of the bread is going to be affected, and it won't. If the quality is affected, the bread formation, the quality of the bread is also be affected. CO2 concentration has all the minerals to a certain extent on the lower side, on the minus side, except sodium Rest of them are a little bit on the minus side. So that will have some implications unless we manage with nutrient management. You could manage it by adjusting the growth, adjusting the adding nutrients to the soil to upset that one. But if they are grown in the under optimum conditions, there is some sort of a imbalance or some sort of imbalance in the nutrient concentrations in the tissues or in the grains for plants grown at elevated CO2. That will have some implications not only for humans but also the animal systems and also on the insect systems, other insects. The insects are also part of the animal group that also has implications for other aspects. As far as the temperature is concerned, drought is concerned, the nutrient concentration, mineral concentrations are not that much. So they're not much change. Only the CO2, all of them are on minus side. Starch is a little bit more, protein is a little.
Okay, sir, proceed.
I just wanted to explore a couple of more things, like exploring about the variability in crops, something like genotypes, variability, genotypic variability. This is for about 40, 45 soybean varieties that we looked at for pollen germination kind of things. There's very small variation, the maximum temperature, optimum temperature, or minimum temperature among those varieties. There is about 45 varieties we tested. There's very small variation. That's not going to make any break or help to a certain extent. We are recently funded with one of these national organizations. It's called a NIFA, that is funded our research, looking at developing varieties for future CO2 levels, future changes in drought conditions, changes in temperature conditions, both high and low temperatures. We have been looking at about 250 different panels, different lines, and then looking at some variation is there, but it's whether that makes that much difference. They may provide some genetic material to breed new varieties in the future, but we are working on that one, those aspects as well. The CO2 response, something, this kind of facilities are needed to look into those aspects, because we can control CO2 concentration in these chambers at specified levels. And also we are also looking at drought conditions as well. Something like a normal condition, a drought condition, and then look at those varietal changes. So in the next couple of years, we will be able to identify some genetic loci that are drought tolerant, or something like genes are responsible, kind of identify the genes that are in those for drought tolerance or CO2 responsiveness or low temperature responsiveness in those lines that we are working on it as well. There is some variability in the commercial varieties. Some hybrids are doing a little better than at high temperatures than normal ones. Hybrids are looking a little bit better than the normal ones. So there is maybe selecting some quality hybrids may be a added advantage in rice. So in order to have some sort of conclusions from this one, climate change has some implications on agriculture production systems. So the one way to is freeze the climate change. Except CO2 is, if CO2 is not going to be associated with other things, that is good for the plants, particularly for C3 plants. It may not be good for C4 plants. C4 plants are much more productive in the current environment. So what has happened in historical evolutionary terms in millions and millions of years, we are making them a little bit irrelevant in a matter of maybe a couple of 100 years. So we are making the C4 plants maybe a little bit less productive at higher CO2 levels. That makes a huge difference in the ecosystem carbon and water balances. Because already the tree species are encroaching these C4 dominated grass ecosystems because of increase in CO2 concentration currently. So that will have some implications to a certain extent. So the other one is focus on increasing the production on existing farms and closing the yield gaps on underperforming lands, and enhance the crop and resource utilization efficiency, encouraging shifting of diets. It's not easy. If you say that don't eat meat-based diet, people won't listen because they got money, they can buy whatever they wanted. So it's not easy to change the minds. Reducing the waste is one thing. 1/3 of the food that is produced is lost due to some sort of waste at different places. Starting from farm to consumers. At this point, we are wanted to utilize all the available technology, including management strategies, to enhance the crop productivity. And there is another issue that is also I'm going to point out a little in the next one after going through. Is now there is a lot of push natural farming, zero input of chemical fertilizers, organic farming. Yes, there are some issues on many aspects. What we see, some of them is particularly if you look at either rice or cotton production in India and other countries, the population, not optimizing population, and then inputting more fertilizer for fewer number of plants per square meter. That's one of the problem we are seeing at many, both in rice and also in cotton. Countries that adopt poor population stands, poor or sparse population, their yields are much less than countries that are adopting higher populations, almost like 10 plants or 9 to 10 plants per meter row compared to 2 or 3 plants per meter row. And then in India, the per pound input of fertilizer is much higher than any other countries. Instead of addressing that one, we are going on some other natural farming and other things that are only successful in certain areas, not for bottom line for across the board. I know there are a lot of push in that one, but it is not easy to sustain. We don't have enough material to put on all the farms that we have. Either we go with organic farming or other forms of systems. So the other point I wanted is if we do not fully understand the long-term effects of human-induced changes on climate, we should not mess with it or refrain from interfering with it. Currently, it is really very difficult or challenging to predict the long-term consequences of climate changes on food production systems. Even though the models are there, but still it is a very difficult task. We must pass on the planet we received from the previous generation to the next, ensuring it is at least in the same condition and quality. That is our responsibility as we move along in this one. Think about also this 2050, the population will exceed 9.8 billion, which is only 25 years from now. And by 2100, it will surpass about 11.2 billion people in a climate that is much different, that will differ significantly from today's if we continue to do the same things what we are doing now. We must produce sufficient goods and maintain sustainable ecosystem services.
No cross-talking, please. Sir, proceed.
We must produce sufficient goods and maintain sustainable ecosystem services for everyone's benefit. So that's what we have to think about it. So for all the work that we have, I want to thank the
ట్యాక్స్ ఏం చేద్దాం అది డిస్కస్ చేయాలి కదా అదన్నా నేను అడగనా ఉన్నారా ఒకసారి
Madam, no cross talking, please.
మంజులక్క
oh, you have a question?
ఇచ్చే
Sir. Thank you, sir. Excellent presentation. That you have stressed the importance of climate change, that carbon dioxide, temperatures, UV radiation, drought, floods, night temperature. So how they are affecting our C3 plants, C4 plants. So your caution that yields must be doubled by 2050. And when the population rate is increasing 2.4%, the yields are 1.2% only. And you are cautioning the society and how they are affecting that in C3 plants, C4 plants, the climate change. And you also stressed the carbon dioxide importance of increase of carbon dioxide, and which is beneficial to the C3 plants and then the C4 plants. And you have also mentioned the countries closer to equator is less adaptable, and open top chambers. You also hook production chambers, sunlit plant growth chambers, and how the cotton yields are decreased and the by affecting of temperature and carbon dioxide, and the how the C3 different.
Oh. I clicked the wrong button. I'm sorry, I missed. Can you hear me? Hello?
Yes, sir. There is a problem with network. I think so.
Oh, I'm thinking of stop sharing my slides. So okay. Okay. I'm sorry it took a while, and there are quite a bit of push in recent years going towards what we call it as a nature farming with less chemical fertilizers, maybe going for organic production compared to the chemical fertilizers. I know there are some issues are there on the I guess the quality of the produce that is being produced, that the food that we eat. But if we can't produce enough food, there is not enough land to bring into cultivation. We already bought most of it. There is not enough. There is no additional land to bring in, except in some areas like in maybe Brazil, Africa, if they get a little bit water kind of thing. But there is no land to bring in. So we have to increase per acre productivity. So if we adapt to other systems, for now, that's not easy. Other systems without, I know that sustainability, environmental issues, all sorts of things are there, but it is there is not enough organic material to spread on the crops, all the cropland that we have. Those are some of the issues that we are facing. I'll be glad to answer any questions related to this aspect. I don't know whether I got lost or not, but if you have any questions, I'll be glad to answer anything related to this or any other related issues related to education or any other things.
Sir, I have a small question, sir. Sorry, in
Yes.
open field conditions, it's not possible to increase the carbon dioxide. We cannot manipulate the carbon dioxide level. But what are the ways of manipulating the carbon dioxide conditions under protected cultivations for higher yield or biomass?
Well, protected yields, they're going to get beneficial. So there will be some sort of cost associated with increase in CO2, unless you have a factory where it produces CO2 and you can construct a greenhouse kind of thing. So you can do that one. But the point I wanted to make it is in earlier, if you look at the literature, in earlier experiments, most of the experiments were done in very low CO2 levels. At that time, it was ambient. 1950s, it was 315 ppm. Today is 425 ppm. So from 425 to 700, there is only small increase, even including C3 plants, small increase in biomass. So by putting that much CO2 into this leaky chambers is not that easy. The beneficial effect will
Sir, some power interruption there. For that, and so you covered all effects of temperature, carbon dioxide, and floods, and night temperatures, drought. So whatnot, you have covered everything. The climate changes in C3, C4 plants, C3 plants, rice, soybean, wheat, and cotton. C4 plants, amaranthus, ranged grasslands, and C3 sweet potato. So you have given many examples. Thank you very much, sir. And now, anybody want to ask a question and answers? Sir is a very eminent scientist in the world. So I invite all teachers, scientists, university officers to ask questions. So, Kiran, Seshikaran, your doubt is clear? Hello? Am I audible, sir?
Yeah, I can hear you, but
Yeah. So now it is open for discussion. Nagaraj garu, you want to ask anything?
Sir, very informative lecture, sir. And from my side, only one question, sir. Here in Andhra Pradesh and other states also, we are observing delayed flowering in fruit crops or other this one. Due to delayed flowering also, due to temperatures, increase in temperatures, we are receiving, the farmers are receiving very less yields. Is there any suggestion from your side?
well, the temperatures we cannot control it until you have a protective agriculture systems like greenhouses or something. Even greenhouses, controlling temperature is a very expensive thing. It's not easy to control it. Summertime it goes up really high, more than outside. So there is not that many options to. There is some sort of breeding, but even so far what we see is most of the advantages that we have is something like a breeding technologies, is on herbicide related, insect related, and fruit quality related, self-life related. Those are, you can see it actually, that's the benefits, you can see it, but not on the yield related that much. That much is not there. So overall, there is a management that played a lot more role, but there is some sort of beneficial effects came from breeding program. I cannot say that no, there is a kind of beneficial programs, but when we tested hundreds of lines, there's some sort of a very small variation is there for now. So it is a big challenge from going from 1.2% per year over the last 60 years of that one to in the next 25 years to 2.4%. It's a really, really big challenge. And if you do adapt to other systems that worry about too much on the natural farming or some other farming systems, the yields are not going to be equivalent to what we are witnessing. There is some sort of, it's a less, I guess in a horticulture crops, also it's a little bit of less concern depending upon the crop. The vegetable crops, leafy vegetables, increase in temperature, increase in CO2 is beneficial. There is
So you
no
have mentioned
doubt.
about, the pollen viability.
That's only in flowers related, grain related aspect. Even vegetable crops, a little bit. Most of vegetable crops, like even at those extremely high temperatures, that's what we looked at it. Extremely high temperatures. Those
Yes, sir.
issues are
Since
there.
you are
That's
from,
where some of the issues. Not like 1 to 2 degrees change in temperature is not going to make that much difference.
Sir, since you are from Chittoor region only, most of the mango farmers, they are facing lot of problems with regard to the pollen viability also. Because of the increase in temperatures, they are losing the production also. For that,
I haven't seen anything that protects the pollen. We looked at the hormones kind of things, but nothing helped in that sense. Molecular aspects, they are there that you spray this and then there is some sort of tapetum or pollen degeneration of cells are there that if we spray with the hormones, it protects that one, it will produce good quality pollen. That is not there. So there are not that many options for high temperature stuff if it really happens at that one. So I guess the pollen viability is very short-lived. It's a matter of hours. If it is produced in the releases at around 8 o'clock or 9 o'clock in the morning and it goes dead by 1 o'clock or 2 o'clock. It's very short-lived and it cannot carry over to the next day. So there are not that many options. So one option is a little bit of irrigation. You can provide irrigation at gets cooler temperatures, canopy temperatures. That's one. If you have irrigation, you have to apply it during flowering time. That's a very good option to have it. If you don't have it water, then you cannot do a whole lot.
sir, irrigation, that is irrigation above the canopy level, will it affect on the pollen viability?
Well,
Above
it affects the canopy temperatures. If they lose water, then it gets cooler temperatures. Canopies get will be little cooler temperature. If they don't lose water, if they don't have water, then the leaf temperature is going to go up, and then the flower and other things temperatures are going to increase a little bit more than the ambient temperatures. So to a certain extent, irrigation during that time period, you have to manage it nicely in that time period. Particularly the pollen viability is very short-lived. It's at 9 o'clock to 10 o'clock, 9 o'clock to 1 o'clock. That's a very short period for many crops. So getting cooler temperatures, providing irrigation is the only time during the flowering time period. Once the fruits are retained, then the size is going to depend on the temperature and also drought conditions, but the retention is the key. So if you have water, that will be the ideal time to irrigate it.
Thank you, sir.
Thank you, sir. Anybody from the student community, scientists, teachers?
Sir, very good morning, sir.
Tanuja, please.
Yes, sir. Sir, thank you for your very good presentation. Actually, I have one doubt in case of carbon credits, sir. Is there any leads in your institute about the carbon credits, sir, for mitigation of the climate change?
Well, there is a, I guess the carbon credit is how much carbon is put in the soil. That's one. So there is a variation among the crops. Particularly, the grass species has more carbon they put in the soil than the dicot species. Typically, grass species they put more carbon. The other one is in order to get a credit, the soil should not be disturbed for a certain number of years. So if we disturb again after 10 years, everything releases back into the atmosphere. So giving credits and guaranteeing that that so and so person or the farmer is going to continue that practice is a kind of challenging. Particularly if we have small farmers, it's very challenging. Whether he's going to continue to do for next 100 years or next 50 years or 10 years or 20 years. That's the challenge, that getting carbon credits. So who is going to guarantee for the small farmers? If you have a large farmer, at least they will practice it, but small farmer like 1 acre farmer, no guarantees that he is going to practice the same thing next year. I'm sure that the carbon credits, you can, certain crops will have a higher carbon partitioning to root systems, particularly grasses have a higher potential compared to the other dicot species to a certain extent. But in order to give somebody, a company, I wanted to buy carbon credits, I want to pay this farmer this amount of money, then what's the guarantee that farmer is going to continue for that one the next 50 years? If it disturbs any time during this time, everything will be released into the atmosphere. Plowing or
Okay.
other things, kind of things.
Thank you, sir. Thank you.
Thank you, sir. Thank you, Tanuja. And almost 3 hours time is over. Sir completed in 2 hours. So it's a very well discussion and almost I think 1 o'clock night.
I'm okay, but we typically sleep late, but that's a little late, but
yeah.
once we pass
So
that one.
excellent presentation, sir. If nobody is there, we'll close it with a vote of thanks.
So one thing I wanted to say for the students, if they are interested to go study abroad programs, there is quite a bit of different countries, countries, particularly Nepal is one of them, Sri Lanka is another one. They have an undergraduate research programs. So with the undergraduate research programs, they have 1 or 2 semesters, the research component is there in the undergraduate. And then they started to publish those aspects, those papers that are either high quality or normal quality journals. When they do apply for, they will get the additional benefit of that one, having a publication or something like that, that experience and having a publication, not only for faculty, but also the students get that benefit. So when I have a selection, wanted to select a student, typically I go with that one. So that's one of the suggestions that Sri Lanka has that one, Nepal has that one, Bangladesh has that one. I don't know how many Indian universities undergraduate programs have undergraduate research programs that gives credit to the students undertake as a research problem.
Rajalakshmi garu, you want to talk? Hello?
Sir, no, sir.
Okay. Thank you, sir. Definitely, it is a exposure. If any student or teacher, scientist want to contact sir, I will provide a phone number. And we have conducted 23. This is the 24 Global Connect series. So if any faculty, scientists, or students want to go abroad and for their study, UG, PG, PhD, postdoctoral, so they can contact us also, contact me also. We can provide phone numbers of eminent scientists working in USA. Thank you very much, sir. If nobody is there, we'll close it. And almost 2.5 hours our time is over. And thank you for taking a lot of pain and excellent presentation. And how the climate is affecting our horticulture crops, food crops, grain crops, corn and all, soya bean, rice, wheat. So you have covered not only that amaranthus, sweet potato, and how the climate is affecting. It is a caution for the society that how the climate is affecting and carbon dioxide level, temperatures, and night temperatures, especially floods. So you have covered everything, and definitely it enrich the knowledge of scientists and students. And I thank you once again, sir. I also thank all our associate deans, all heads of research stations, and all university officers. And I thank profusely our Honorable Vice Chancellor, Dr. K. Gopal Garu, for giving permission to conduct this Global Connect series. With his encouragement and support, we have completed 24th Global Connect series to enrich knowledge at what is happening in the international arena, by arrange guest lectures from the eminent scientists. And I thank all university officers for actively participating in this program, and make it a grand success. And I also thank all the teaching and student faculty, students, MSc, PhD students, UG students who are participating in this program. Thank you one and all. Thank you, sir. Thank you very much. Have a great day.
Yeah,
Thank you.
thank you.
Thank you, sir. Now the program is closed. Before closing, just I want to have one reminder that last Zoom meeting with all engineering institutes, Sasi Institute of Technology, Vishnu, NIT and WIT, and West Godavari Institute of Technology, engineering institutes, we have given some projects, collaborative research projects as directed by the Honorable Vice Chancellor and all university officers. And some of the institutes have sent projects. Most of the institutes have not sent projects. Kindly, because we are going to sign MOUs with the above institutes second week of June. But before that, if you send the projects, we will finalize it. And as directed by Honorable Vice Chancellor, kindly send the projects you prepare immediately and send through proper channel to the DIP section. Thank you, sir. Thank you, one and all. Now the program is closed.
Okay. Thank you. Thank you for the opportunity, and I'll.
Thank you, sir.
Yeah. Good night.